So hi, hello and welcome again. Microbehunter here. Welcome to another Saturday microscopy live stream. Oliver here, and I'm happy that again many of you joined in. I've uh, prepared the following for today. I've got here 8 microscope slides. Some of them are pretty old, over 100 years old, and I would like to look at them with you together today. I see that many people are already have already joined the chat forum. So I would like every week, I would like to ask you if the sound is okay, and because I do not want to talk and then you're not able to hear me. And if you're new to this format here, please do feel free to ask questions in the chat forum. I will try to answer them as long as they are microscopy related. But please do indicate if you have a question, and please make an @Microbehunter or an @Oliver sign, so that I know that the question is directed for me, because sometimes there is quite a bit of communication going on between the members, and therefore it's easier for me to find it this way. Okay, so yes, I see that usually during the first couple of minutes I do a little bit of small talk, because it takes 3 or 4 minutes for people to join into the live stream, and usually at the beginning here I say hi around the world here. So hello from Vietnam, from Bodensee, Germany, from Rhode Island, United States, okay, North Miami Beach, Florida. Somebody even in German. Ich bin aufgeregt für diesen Livestream heute. Yes, someone is very excited, so am I. And sound and stream are okay. That's very good, because maybe you've seen I've got a new microphone here as well. And if for whatever reason the live stream is interrupted or cut off or breaks somehow, it happened some time ago, then please be patient. I will try to restart the live stream, but it might be that it will be in a different link, so you have to check the YouTube channel again. Okay. So I would like to give you first of all a little bit of background here. I bought those 8 slides because that is a question here. I got them over eBay. And I basically selected them based on personal interest and whether they look nice. Yeah. I would like to look at them one by one. And as a matter of fact, there's already one over here. And some of you might speak German, and you might or might not be able to actually understand what's written on here. I was actually able, with the help of a very unlikely person, I was able to help to decipher the text here on this slide label. This very unlikely person, unexpected person, is no one less than my own mother, who is still able to read the old German handwriting. It's known as, in German, it's known as Kurrentschrift. And the letters are all different and so on. But I was able, I basically, there's lots of information here, and I basically was able to figure out what it is. Okay, so we'll be looking at this as well. And there are already a couple of questions here. One question is, what materials did they use back then to prepare and preserve the specimens? And you'll be surprised by the answer. Sometimes very similar materials as we have nowadays. The mounting medium, for example, is even indicated over here. It's Canada balsam, which is a very, very reliable mounting medium. So if the specimens are prepared well, the slides are in good shape for over 100 years. But not so easy to use. And other more modern mounting media have replaced this. But I've heard that some people are still using this one here. And there's the staining is also mentioned here. So I will be explaining all of this. And essentially, because I only have got 8 slides right now, I don't know how long it's gonna take me, so maybe I'm going to run out of things to cover in this live stream. So if you ask questions, then it helps me to fill the time a little bit. Okay, so this is basically we'll be looking at those one by one. And another important thing is I'll be making a separate video for each one of those slides that you can watch in the channel. And as a matter of fact, maybe some of you have already looked at my most recent video, which I published just a few days ago, which is this one over here. This is actually, that's a very unique specimen. This here is the finger of an unborn baby. Okay, it says here the section of the finger of a 4-month-old fetus. And yeah, so I already made a separate video of this, which you can watch in this channel here. But of course, we'll be looking at it again. Okay, for those of you who have not seen it yet. But I will make a separate video for all of those here. So what I'm going to do is I'm going to start with I would say the most difficult to identify slides, and these are the ones with the orange label here. The text is, as I already said, I've got 3 of them written in German. These are the slides with the most information on them. But because it is written in old German handwriting, it can be quite difficult to understand what is written on here. Okay, so this is actually where, as I mentioned, I needed the help of my mother. And I'm going to do the following. I've got 3 slides here, but I actually made pictures here. I would just like to show them to you. Here, I printed them out, and I added, of course, also translations here, so that so that you know what we'll be looking at. Okay, so yeah, here are the slide labels in big again, and that is actually on the left side. This is actually says something about the preparation technique. Yeah, so it's basically the staining is hematoxylin and eosin. It's a very common staining. I do not know what this character means here, honestly. So if there's any one of you who is familiar with old German handwriting, maybe you can help out here. But also my mother was not able to help me out on this one over here. I thought that it is an old way of writing an and sign, hematoxylin and eosin, but no, it's not. Okay, this is the mounting medium Canada balsam. That's quite clear, and this is the date. It's the 20th of January 1923. So this slide is just over 100 years old, and this one means number, but there is no number written over here. Okay, it is according to the information from the seller. It is from the University of Stuttgart in Germany, Mikrokosmos Stuttgart. Yeah, and so that's basically on 2 of the 3 slides I was able to find this information here. Okay. So and then on the other side, I just want to show you. There's also quite a lot of information here. So this one over here is there's a Latin name here, Aesculus hippocastaneum. That is basically the horse chestnut. Okay, Rosskastanie. So this is basically the German word of the horse chestnut. I added the English translation here. This one over here says Fruchtknoten, which is the ovary of the horse chestnut. Quer, it took me quite a bit of time to figure out what that actually means. Quer means cross-section. And this seems to be the name of the person here. And then, the writing over here, my mother told me this actually is very different. This is the modern writing compared to this one over here. This is the old German writing, but this uses modern letters. So she actually said is that this must have been from a time where there was a transition in writing, or somebody else actually assigned it. This could also be a plot, which I don't think. But in any case, you see, the lesson that we can learn here is it's a good idea to include as much information as possible on those slides. But again, if it's difficult to read. Yeah. So this one over here is another one. Acer campestre, that's an ahorn. In German, it's a maple tree. Again, it's the ovary, the cross-section, and over here, and again, the name of the person. Okay. And here, the third slide here is a flower. Iris sibirica, in German it's the Schwertlilie, Siberian iris, it's a monocot flower. Frucht, again the ovary. So these seem to be botanical slides, cross section, and again down here these are the ovules that you're able to see here, Samenanlagen. Honestly, I would not have been able. I've got a biology background, so I was kind of able to make sense of the words here. But people without any biology background, for example, I was able to figure out what it actually means simply by these with A and L, anal, and this actually, and A, so I could already know, it must be Samenanlagen, which are the ovules, right? So you see, even I had some problems here, obviously. Yeah, this, by the way, this is the flower that we'll be looking at. That's how it looks like. I got this from Wikipedia. It's a beautiful flower, and that's basically the cross section here of the ovaries. Okay, so this is basically these are the slides that we'll be looking at, and I will be basically just going through the slide box a little bit, starting with those slides here, and every now and then I'm going to interrupt myself, and I am going to also look at the questions again. I'm curious, how long do you think the PVA glue slides would last? Do you think they'd be lasting as long as those slides? I don't know about that. I know that the PVA, polyvinyl alcohol or polyvinyl acetate, that is indeed a water-based mounting medium that has been used quite a lot in the past. But I don't know how stable the glue is. If it is properly dry, and if it is not exposed to much humidity change, and I can imagine that it will last. But yeah. So this one over here is the horse chestnut. Okay, and when we look at this, we can actually see a little bit here. There is already a little bit of a discoloration of the mounting medium here. Okay, yeah, this is quite common in some older slides. It could be that maybe because of the oxygen in the air, maybe it starts to oxidize a little bit. Yeah, so as you can see, it kind of it starts to discolor a little bit. But let's have a look at this slide first. Okay. Yeah, so and then I will simply remember where I stopped off with the chat, and here is now the microscope view. And again, some of the samples are more interesting than others. It doesn't matter, it's still quite nice. Okay, so of course I always have to, I have to focus again. Here we go. Okay, yeah. And we open this, you know what, I just open this a little bit better, and I remove the filter. Okay, here we go. Now you see that this is a little bit cut off here for whatever reason. But what we're able to see here are already 3 ovaries and with the ovules in them. So basically this is the whole structure that you see. You know what I'm going to do is I'm just going to turn on the I think the whole structure here, that is known as the ovary, and in the ovary you have those ovules, and those ovules will form the seeds, and the outside here, the ovary will form the fruit. So in this case, ovary is usually a term that you associate with mammals, but no, in botany it's also called ovary, and so that's basically. And I'm going to now always start to focus it. That's a little bit annoying because I have to reach over to my microscope, and let's start to zoom in, and let's not forget that this slide here is over 100 years old. Okay, and this is how it looks like. Now I've been kind of wondering a little bit what those brownish looking structures are. You know, obviously there must be somehow cells, but but they look quite different from the others that we have here. Okay, so and it's interesting. Normally what I would have said is that interesting, I find those outgrowths here on the side a little bit interesting here. These are usually, I mean, normally if I would see those alone, I would say, these look like side roots, you know, the root hair a little bit, yeah. But this is of course not the case because we're looking at the cross section of the ovary. Yeah? So, and in here we have then basically the part that will actually form then the seed. And if we zoom in yet further, and I was not able to do that, but I do have a couple of other slides where I was actually able to see also some of the cells dividing, but I was not able to find a dividing cell here yet, at least. And what we see quite clearly is that the nuclei are stained. See, I always have to refocus a little bit here. Okay, and so let's go up a little bit. Yep. Okay. So of course those dark structures in the cells, these are of course the nuclei. That's where the DNA is, and you can also see of course the cell wall on the outside. Okay, so those lines here, that they kind of define the cell. You're going to notice now that some of the cells appear not to have a nucleus, especially over here. So why is that? Well, of course they do have a nucleus, but the cross section is so thin that the nucleus was actually cut away. Okay, so it's either above or below this cell because the cell is a 3-dimensional structure, and therefore, it's simply the cut simply missed the nucleus, so to say. So this is simply And the space also tells you a little bit something about the size of the cell. So if the cell is very thick, then of course the probability of actually cutting the nucleus away is of course higher. That's why those cells here seem to be not only a little bit less dense, but also seem to have less nuclei. It's because they seem to be generally a little bit larger here. Okay. Yeah, so here we are again, and those, honestly, this, I don't know what this is, okay? What these cells are, why they look the way that they do, okay? That is something, those cells here, they have a very thick, seem to have a very thick cell wall, okay? And this is actually an indicator that this could be the so-called xylem tissue, because xylem is, these cells have a thick wall and they transport water up a plant. So this is something that I suppose that we're able to see here. So I'm currently using a so-called DIC. Okay, but if you were to look at the whole thing, so the whole thing, yeah, you see I can also change the color around. But if I remove the DIC now by flipping out the filter, then this would appear in bright field like this. And these are now basically the natural colors that we see. Okay. So, I have to tell you, I'm 100 years old. I'm very happy with the quality of the slide. I mean, of course, modern slides also can have a very high quality, but I've seen some modern slides. There was so much dirt and so much contamination because evidently the slide was not cleaned. Right? But it's quite different here. Usually, by when you're looking at the place where there is no specimen, and you see, of course, you see a few things here, but generally it looks fairly clean. Huh? Of course, from the preparation, there might be a few, dust grains here, but generally the specimen is quite clean. So the person who actually made the slide put quite a bit of work into this. Yeah. So I really like this. So what I generally would say is that making those plant cross sections. I mean, that is you can find a lot of modern slides that have those. That's not so the biological significance here is not really that high. I mean, it's not that rare. Let's put it this way, right? I mean, this was a wrong way of saying the biological. It's not biologically not rare. The specimen, it's not a rare specimen because anyone can prepare that. But I think that what makes it special a little bit is simply to know that over 100 years ago, somebody put quite a little bit of energy and work and maybe also even a little bit of love into making those slides. Yeah, and that's why we have them now. So what I have to say is the following. If you are also into preparing microscope slides, permanent microscope slides, do add a proper slide label with your name and a date, because who knows, maybe in 100 years, 200 years, somebody's going to rediscover your slides and then maybe they're also going to be sold on eBay. Yeah. But you see, generally I like looking at these things, and occasionally what I've done in the past as well, if I see something that I kind of like, I will then basically just take a picture of this and frame it and so on. So it's simply for the aesthetics of it. Yeah, yeah. It's a little bit, comment here, 100 years old, it's something we should appreciate today. Yeah, I mean, honestly, really, the person who made the slide, it's one thing I can absolutely guarantee you, the person who made the slide never would have, in his wildest dreams, would have imagined that 100 years later, somebody's going to, hundreds of people are, thousands, over 1,000 people are going to watch this live stream after some time. 1,000 people are going to watch this preparation. Yeah, yeah. The comment here, I'm impressed that they could get such precise cross-section back 100 years ago. Yeah, because apparently microtomy, the ability to cut very thin sections, was actually already developed at that time. I don't know how it was embedded, if the person also used paraffin embedding, yeah. But I mean, this was in the 1920s. Yeah, here again. Yeah. So again, I do not see any dividing cells or chromosomes here. Maybe I'm overlooking them, but in any case, I have some specimens where indeed this is visible. Yeah. So there is I'm going to quickly do the following. Yes, there are a couple of very interesting. Have you ever tried to use UV nail polish instead of UKIP or UPROL? I bought it and used it and it works great for me. How long will these slides last? What's your opinion? So first of all, that's an interesting question. Now, I know and I have heard of UV nail polish, and I have not tried it out yet, but I do know that this is also not a new idea. UV curing of microscope slides, this concept that does exist. But I have not tried it out yet. And my answer simply is the following. I don't know how long it will last. Generally, a lot of the stability depends on maybe not only on the mounting medium itself, but on the actual preparation of the specimen. So you have to make really sure that the specimen is completely dry. That's really important. And you have to give it a little bit of time also to the specimen to absorb the mounting medium. So what I probably would suggest is that when you place the specimen into the UV nail polish, do not shine the UV light on it directly, but maybe give it a little bit of time, and possibly also something that depends a little bit on the specimen, do try to apply a vacuum, because what happens is that all this will remove all of the air bubbles, and it will allow the mounting medium to infiltrate the specimen better. Okay, so what I'm trying to say is I know I didn't answer your question directly because I don't know how well UV nail polish, how stable it is. But what I can tell you is that a proper infiltration of the mounting medium in the specimen is quite important. Okay. So I bought a few boxes made in China, sample slides about 25 years ago. I'd say half of the specimens have deteriorated. Oh, really? Okay. All my antique slides are still good. Thank you for the information. It's quite interesting. Yeah? So, yeah. I'm trying to look at some dead coral under the microscope, so I took a small piece and put it on a little bottle cap, but I need some help since my microscope is not a stereoscope. Okay, so if you want to look at specimens that are very large and you do not have a stereo microscope, then actually what you can do is the following. You use the low power, and that's more than enough, okay? And you place the object directly on a blank microscope slide, and then you look at it and you use a strong flashlight from the top. You'll be surprised what you're able to see, okay? So even if you want to look at small coral pieces, I have done this as well. I've also made some videos even about using a compound microscope with top illumination. So it is possible to do that, just make sure that you've got a good flashlight. Okay, so what I'm going to do now is I'm going to change, because I'm going to now move on to the next slide. Okay, so this one over here was the chestnut, the horse chestnut. So let's put it away. So let's try the next one, because I have a couple of beautiful insects here that I want to show you. Okay, so let's do the plant anatomy first. This is now in Germany called a Schwertlilie, Siberian iris. It's a flower. And again, what we have here are the ovaries. Okay, so they are going to form the seeds and the fruit. Okay, this actually looks also quite nice. I'm going to show you the sample. There's 3 of them here. The sample is a little bit more strongly stained. Okay, and let me center it again here. Okay, I'm way too bright, of course. So I'm going to go down here. That is how it looks like. Okay, so that is again the full ovary, and in those dark bulb-like structures, these seem to be the ovules which will form the so-called the seeds. Okay, so here again. Okay, unfortunately there, as you can see here on the top, there is the left slide label is missing. I also do not see any leftover glue or anything like that. And it is also not signed the same way that the other 2 slides are, so it could indeed be made maybe by a different person, I don't know. But in any case, so these here are again the ovules, basically, which will start to form the seeds, and the whole structure here, that is the ovary. All of those little dots that you see are again the nuclei, lots of cells. So I mean, you might already know that I'm a biology teacher in my non-microscopy life when I'm not making YouTube videos, and of course I do microscopy also with my students. I remember this one case when we did some microscopy like this, and for the student apparently it was the first time, and all of a sudden she was looking at one of those specimens. I remembered with onion cells, the totally standard thing, right? And then all of a sudden she looked totally. Yeah, exhausted and frustrated, and it's okay. Again, she had this problem with a microscope. So how can I help you? What problem do you have with a microscope? And then she says, no, I have no problem with the microscope. I'm just so impressed. There are so many details. So she was totally, blown away by the fact that I could not imagine that in just an insignificant piece of material that you're able to see so many details and so many cells and such a high degree of complexity. Yeah? So that's actually that's the way it should be. Yeah. Sometimes when I mean I'm doing a lot of microscopy, obviously, you kind of start to. There's always the danger that you start to kind of realize, of course, it's another plant cross section. But in reality, you just look at the complexity of everything here. So that's over here. This seems to be definitely some kind of a vascular bundle here, where water is transported. Okay, so probably that might be the xylem cells. Yeah. Again, you see all of those little dots here are individual nuclei. Yeah. So. Also here, the reason why I think that these are the cells that transport water is because, again, they have a fairly thick cell wall, okay? Because they are usually also strengthened to support the plant. Again, over here, the ovules, which will start to... here it is. The ovules, which will start to form the seeds, and it looks very dark. So this actually means that there are lots of cells clustered together because the stain evidently stained the DNA quite well, okay? So here it's still fairly thick. So actually getting it well into focus is difficult. So let me open up the condenser a little bit here. So that's a case for a little bit of focus stacking. Yeah, but you actually see that when you actually operate the fine focus knob, how some cells actually start to move into focus and others go out of focus. Okay. So again, I'm going to look at a few questions. So is there any way to turn my compound microscope into a stereoscope? Well, not a direct way to do that. But as I mentioned before, if you use the low power and if you use a flashlight from the top, then this is going to give you a very good impression. And there's another thing that I don't know if I have it here. Just a second. I don't know if I have it here. No, I don't seem to have it here. But there are those cheap USB microscopes that you can buy. Yeah, don't spend too much money on this. But if you do not have the money to afford a stereo microscope, buy yourself those cheap USB microscopes that you can just put on top. The quality is not quite as good as a stereoscope. Obviously, you don't have a stereoscopic view, but you can do also quite a bit of casual exploration. Okay. Do you have a Discord or plan on maybe make one in the future? Well, I've considered it, but the thing is, it would require me to also maintain that, and it's simply a time issue that I have. Yeah, I do have. If you go to the webpage microbehunter.com, then there is a forum there which is pretty active. You can sign up for that, but please do send me an email. Otherwise, I'm not going to unlock you because I'm getting so many spam registrations. So you have to register first, and then if you send me an email with telling me that you've actually registered, then I'm going to unlock you. Okay, but I do not want to make this too automatic because I've gotten so many spam registrations. Yeah. So, okay, I'm going to go down here. Lots of comments. Okay, so what I'm going to do is the following. I'm going to go down a little bit with the magnification. I'm checking the time. Oh, I'm already talking half an hour. Okay, good. Well, I was actually worried that I'm going to be too quick with everything. So let's move on. So this was slide number 2. Let's move on a little faster because I do want to show you the ones from the insects because that looks really nice. So this one over here is, what is this here? This is Asa ahorn. It's the maple. Okay, look, it says here, yeah. And again, a cross section by a person named Veda. So I don't know, honestly, I don't know, maybe someone of you has connections to University of Stuttgart. Then I would be very appreciative. I would appreciate it if you could maybe you were able to figure out who this Mr. or Mrs. Vedder was. Okay, I think this one is not such an exciting one. Okay, this is a little bit one of the more boring specimens, but again, it's the same example of a cross section of an ovary. So I again have to make this a little darker, and it's a little bit more difficult to find now. Okay, but evidently these are all botanical slides. Okay, that's the side of the here it is. Okay, despite the fact that the plants are different, okay, there is still a certain similarity here. Again, the ovary, the whole structure, the ovary, and on the inside here, you have the ovules, which will form the seeds. Yeah. So over here we go again. Okay, and 40 times, and here, up, I bumped it, and we open the thing again a little bit. So let's go up to 60 times. This is the working distance. It is very close. Okay, so sometimes it actually bumps into the So the arrow. I'm gonna move the arrow out of the way a little bit here, and I just also want to show you a little bit, just for the sake a little bit, because some of you might not know this. This is my 60 times objective, but notice that there is a scale over here, right? And this actually is for the cover glass thickness. And by turning, there's a ring here that you're able to turn. By turning the ring here, you're able to optimize the objective for the best resolution. So maybe it's a little bit blurry now, right? So this is actually for adjusting for the cover glass thickness. Some objectives have that, especially if they're very high resolution objectives. Then the thickness of the cover glass actually does make a difference, and this allows you to correct for the thickness of the cover glass. Just wanted to show it to you in case you haven't seen that. Okay, yeah. So let's have a look here again. I'm going to do the following. I'm going to now rotate this the polarization, and sometimes you're actually able to see those little things here. Sometimes there are indeed crystals inside the sample, but I think that this is probably more of a contamination what we were able to see here. Okay, could be crystals, but could also be some kind of contamination. Sometimes you have to check the side now, to what extent these are also on the side. That's the next specimen here. So if you look at crystals, they look a little bit more regular and can be found more evenly scattered about. Okay. So we go on here again. Okay, so again, this one here again was stained with hemodoxylin. It does not say that it's stained with eosin. And it has the same date, January 20th, 1923. Okay. So we're not professionals. What do you choose to show us? Okay, this basically what we see here. Maybe I have to be a little bit more basic with my explanations here, because I assume, with my talk that there's a certain that you understand already my terminology. But of course, if I say ovary or ovules and so on, then of course this is not understandable if you're new to the topic, of course. So thank you for reminding me here. So this here is essentially a maple. From a maple tree, in the flower of a maple tree, this is where the seeds will form. And this is a cross section of this, and this is known as the ovary of the flower. So this is inside the flower, and in here will be the ovules. These are the egg cells of the flower, which will form seeds. So this will actually form a so-called a fruit, and inside here there will be the seeds. Okay, so this is kind of the idea. If you just joined the live stream, the main idea of this live stream is actually not to show you botanical specimens, even though I'm doing that now, but actually the main idea is because I wanted to show you very old microscope slides which I bought, and this microscope slide up here is over 100 years old. So that's actually the main point here. It's not so much of giving a biology lesson here. Okay. But I would like to move on because you probably want to see also some other slides. Okay, so what I'm going to do now is, this is I would say, the one that I'm most excited about. Okay, and this is the one that I already made a video, so in this channel. So if you just joined in, this here is, I would say, quite a thing here. This shows this. It's labeled now in English. Section finger of fetus 4 months. So this is a finger of an unborn baby, of a fetus, a cross section. There is a number 18 written here, and I assume, I don't know, but I assume that this is basically for writing the date, 1800 something. And you're supposed to fill in and complete the date. Unfortunately, it's not been completed. And I'm just going to show this to you very quickly. And if you want to have a little more information about this, I actually, of this one, I made a separate video just a few days ago. Just a few other interesting things here. You notice that the slide is ringed. Okay, so this has several functions. The ringing, first of all, it will stabilize the cover glass. So that means so that the cover glass does not separate so easily. It can also prevent oxygen and moisture from entering the specimen. And a third reason is sometimes people do that for beauty, for aesthetics reasons, because it kind of looks nice. But you notice already one thing, that actually the paint has come off quite a bit here on the surface. And if you turn it around, the slide, then you will see that there is also some of the paint over here on the edge. Okay, here, right here. This is from another slide. So the fact that the paint has come off here actually shows that apparently the slide must have been simply stacked together on top of other slides. Yeah, and this is why it kind of got scratched off here. Yeah, so I think that this slide has not been treated with a lot of respect. Just holding the slide, it feels different. First of all, the slide is a bit thicker than the modern slides, so I actually had a problem in fitting it into some of the slide boxes. It does fit into this one here because the slots are a little bit wider, but into my other slide box I was not able to fit it, because it's thicker, and the quality overall is also very good because it's polished. By the way, all the other slides as well, they're all polished. Yeah, so basically you're not able to cut yourself, and it feels very of high quality. Okay, so this is really so this is something I'm going to be looking at. Could the number be a sequence number of slides from the same finger prep? So the question, this is an interesting one. The question is could the number over here be some kind of a sequence number? And the thing is the number 18 looks printed. Okay, it looks printed. If I don't know if you were actually to print in slide labels with consecutive numbers, you know. So this is the thing. It could be, but I don't know. I do feel it's a pity that there is not more information on here. I do not know who made it. But I will just show it to you now. And there are a few things that I can also explain to you here. Okay, just a couple of minutes maybe, because I did make a more comprehensive video, as I already mentioned. So we're going to go down all the way with the magnification again. I'm going to flip out this here, so we're going to just look at it using bright field. Okay, I'm going to turn it around because it's the way I'm used to it. I was used to. So where is it? Here we go. Okay. There are a couple of structures that I can actually explain. Not all of them, but a few. You see here now it's in focus. Okay. Yeah. So yes, we are now here at the region of the fingernail. Okay. And I'm going to just, yeah, and if you just zoom in, this is actually one of the regions where the layer is fairly thin and where we're able to again see individual cells. Okay, yeah, so this is basically, yeah, looks like this, but the really interesting parts are somewhere else, I would say. Okay, so let's, I'm going to show you now a few interesting parts. I have to adjust always the light again. So let's start to move up here. Let's start to move over here. And this part here. This here, this brownish looking thing, structure. Yeah, that is, I'm quite sure, that is a blood vessel, and this is these are the red blood cells clumped together. Okay, found in a blood vessel. So this is the blood vessel. You can actually see also here the wall of the blood vessel. Okay, and the individual red blood cells are not visible when the blood clumps together. Okay, and the red blood cells do not look red when the hemoglobin, which is the red pigment, it starts to break down, and then you have this brownish looking, clot. Yeah, and if you look at the side here, look, go over here all the way to the other side. Look, it actually looks like we're able to see the cross section here of the blood vessel, right? And on the other side, when we go all the way on the other side of the blood vessel, we can actually see that it seems to split up a little bit here. Yeah, it's almost branching. So I do assume it's a blood vessel. Yeah? So that's one thing I just wanted to show you. And there's another thing I wanted to show you, which is further down here. And when I saw this the first time, I said, what is this? Yeah? Look at all these horizontal structures here. Okay, so maybe muscle fibers, and then I said, no, there are not so many muscles in a finger. The muscles that move the finger are in the lower arm, and this here, I'm quite sure, must be collagen. Collagen is a protein, and that's the tendon. So basically, the tendon is a very strong tissue that pulls on the muscles, and therefore allows you to move the fingers. And when you zoom in, there is again, if we make it brighter, this looks very much like collagen. Okay, you see those horizontal lines, yeah, and so therefore I'm quite sure that I assume that this basically is the tendon, and I of course also googled a little bit how does tendon look like under the microscope, and indeed you see sometimes those very thin, maybe difficult to see darker regions that are interspersed here, which are the individual cells. So I'm going to read again the whole thing here. So just a second. I've missed a few of the questions here. So I'm going to do this again. What is the best way to store slides besides in a box to make them last 100 years? Constant temperature, constant moisture, and proper slide preparation. And then a little bit of luck maybe. Really. That's a little bit of a question. I know I've seen some microscope slides that are very old and in very good condition. Other slides start to decompose and break down after a very short time. I think a lot depends not only on the storage, but probably also on the proper preparation. Yeah? Is it possible to view these kinds of slides with dark field, or is it something that only works in wet mounts? I don't have good dark field here. I can a little bit. I can show you dark field with the 4 times magnification and using my phase contrast annulus instead of a dark field, and this is how it looks like. Okay, so you see, this is not the best specimen. Dark field works really well. You see actually more the dust in the background, and it's not a good. I don't have a good dark field setup. Okay. Dark field works really well if the specimen has a strong difference in refractive index. Okay, and this is not the case here. So dark field in this case is not the most suitable. So, but there are. Let me quickly do the following. Let me quickly show you a few things here as well. Where is this place? I'm looking for this one specific place. Let me see. Here it is. Okay, here it is. Here we go. We're now at the skin, and you see that sometimes over here there are these little lines, almost vertical lines going towards the skin, and in some places it's more pronounced than in others. Here as well. Okay, see those vertical lines here, and then there's usually some kind of a larger structure here, and I think that these might be the developing sweat ducts. Ducts, basically sweat glands over here, and ducts that are kind of released in the sweat to the surface of the skin. Sweat glands or oil glands and so on. So that's, I think, the thing. Because they appear in a quite regular intervals. So let's move a little bit to the top somewhere. Again, So we see something similar here also on the top. Some darker structures beneath towards the skin, which might be developing glands that will then secrete substances to the surface of the skin. So, would the use and overuse of light also add to deterioration of the medium? So basically, in other words, I will rephrase: will use of too much light result in bleaching of the specimen? And I suppose yes. Because any pigment that there is in a specimen will mean that the light has to be absorbed, and this absorbing of light energy is actually your energy input, and if now this energy is used not only to heat up the specimen but also maybe to cause a breakdown of the pigment, I can imagine that maybe bleaching can indeed be if it's done too much, maybe an issue. But I've seen, however, bleaching also happen, especially in natural specimen. When you look at algae, for example, then freshly mounted algae, permanent mount, simply look much greener than older specimens. So in the case of chlorophyll, with the green pigment, this is indeed the case. Okay. Yeah, so you know what, if you want to know a little bit more about this specimen, I did make a separate video, okay, of this in the channel, and I'm going to now do the following. I'm going to move on because I would like to show you a few more slides. I'm halfway through now, and I'm going to just leave. Where is this? Can we actually see the writing here as well? I'm just wondering. See, that's the writing, the handwriting, and this is the printed number 18 of the slide. So I turned, So you see, this is not handwritten, so this is part of the print of the slide, and this here is the handwriting with ink, and you see that of course the ink is not quite as even. You could actually see how the ink kind of spread over the paper. Okay, just wanted to show this to you as well. Let me quickly remove this here, and desk again. Let me put this carefully in here. So, what is this here? Tongue of a honeybee. Apis. Okay, that's the Latin name, and this is the specimen. Honestly, unfortunately, no date, no information here on the slide who made it. We'll check quickly I will quickly check the eBay thing here, the pear or tongue of honeybee by W. Watson. It says the eBay information says the following: tongue of honeybee by W. Watson. Okay, maybe some research you can do here. Okay, and I think that this is an incredibly beautiful slide. I think this is a really nice, never seen such a nice preparation before, because it's very symmetrical and very complete. Okay, so let's do this again. Scope, view. Okay, and way too bright again. Here it is. Let me focus this again. So I'm going to now move the slide. This is the tongue of a honeybee. And I think really nicely placed. No damage. Okay, look at all those hair. Yeah, so I really kind of like this specimen here. Okay, so let's go up a little bit with the magnification. Okay, so, and yeah, I see that there's still a couple of questions in the chat. I'll try to answer them later. So that here is the tongue of a honeybee. Proboscis. Okay, lots of hair, evidently sensory hair. Okay, let's go in a little further. I mean, maybe some of you, why not? Maybe somebody can figure out and do a little Google search. Who was W. Watson, who made the slide? Maybe a company. Yeah. I think it's really nice. Huh? Let me put on the DIC. And let's be able to increase the contrast a little bit this way. Yeah. Pretty nice. Okay, let me quickly go here. What about fruit without seeds? Well, I gotta be honest with you. The fruit is actually the purpose of spreading seeds. So if you have certain fruits without seeds, then what I heard is it is possible to do that by treating the plant with certain plant hormones, which triggers the formation of the fruit before the formation of the seeds. Yeah. So, but actually, the fruit is there to spread the seeds. Okay. Could the number be a sequence? Okay, that is something I already talked about. Where do you think the owner of this slide got the finger, referring to the fetal finger? And honestly, in the medical field, there are plenty of specimens available. Okay, so yeah, so it shouldn't be anyone who's got some kind of a connection to a hospital. They have plenty of specimens available. Yeah, so it's not really difficult to get those. And how would people collect those antique slides? I'm not quite sure if I understand the question. You basically obtain those slides and then you just collect them. Yeah. So yeah. Could you try polarized light or rather cross polarization? I'm now using polarized light, but it is not cross polarization. Okay, so I have to actually I don't have it here now. The filters. It is possible to do that as well by actually putting the light between two cross polarized filters. Okay, but actually I do. This is not polarized light, but it is not because it's DIC. It's not cross polarized. Okay. So let's have a look here again. So that is, yeah, look at all of those details. I think that's a very pretty looking specimen. But I'm going to give you again a lower magnification view here so that you see a little bit how it looks. And also, I think very nice is the fact that the specimen is not so dark that you're not able to see anything. Very often insect specimens, for example, they are very dark, so you have to bleach them. Oh, wow. Okay. Do you see these things here? Let me zoom in a little bit. It looks like a tracheids. Tracheids. Could this be the tracheids system? Of course. Oh, that's also nice. Interesting. Okay, it's very faint to see, but that is how insects breathe. Okay. So what insects? Yeah, very nice. Definitely. The so-called tracheids. Those tubes here. Okay. These are tubes basically that in which there is air. And they're connected to the outside, and those they branch up and become thinner and thinner and supply the insect with oxygen. And so insects, obviously, they don't have lungs, but they have those system of tracheids which are open to the outside of the body, and this is how the gas is pulled in, the air is pulled in, and this is how they actually breathe. So that's basically their way of increasing the surface area for respiration. Yeah. Oh, look, it's actually opening up outwards. Interesting. No, it shouldn't, because the openings are on the abdomen of the insect. Yeah, but these are basically the way how the bee breathes. And also those ring-shaped structures are for strengthening, so that those tubes do not collapse. So let me quickly. So there's a question if I already showed last week's onion slides that were drying. No, I didn't show them yet, but I have them here. Okay. Somebody found. Thank you very much, Crispy Bee. John W. Watson, 1830 to 1919. So that's probably a lot older than 100 years. Thank you very much. Okay. There is the name W. Watson is not on the slide, but it was on the page of the seller. Okay, so 1830 to 1919. Wow, very nice. Thank you very much for the info. Okay, another info here. Okay, thank you very much. W. Watson and Son was an optical instrument maker in 1837. The William Watson business was established in London for the manufacture of optical instruments. Thank you very much. There were also ads for microscope slides from J.W. Watson from 1861. So yeah, they're really old. Wow, very nice. Thank you very much. Okay, more info. W. Watson and Son was an optical instrument maker. Yes. Slides in the 1870s. Okay. Sir, why does it look so dry since it's a tongue? Well, because it is dry and it has to be dry because it was dried, because otherwise you would not be able to store it that long, right? The microscope slides only store for a long time if the specimen is completely free of water. So actually drying it in a proper way is a little bit of a science in itself, because if you dry it too quickly, then the objects shrink and start to deform. So actually having a correct way of removing the water slowly by treating it with alcohol and so on, that is actually the way that they've done it, or is still being done. But it's really correct. Of course, in real life, those specimens that I all showed you, of course, contained water, because they're living, and living objects contain water, right? But for making a microscope slide, it has to be completely dry. Okay, so So this is very quickly again overview of the tongue of a honeybee. Slide from W. Watson, optical instrument maker. And I guess obviously they made also microscopes, and when you sell microscopes, the people also want to have some slides, obviously, and that's why I'm quite sure that they also made slides so that the people who buy the microscopes can also look at a few things. Yeah? As a matter of fact, in Great Britain, in the Victorian time, 1800s and so on, hobby microscopy was very popular. And somewhat of another curiosity is that sometimes at that time, apparently it was quite common for basically certain educated class of people who could afford it to actually have a microscope at home. Simply because, obviously television and things were not invented yet, and the internet didn't exist. So how did people keep themselves entertained? And some people actually had a microscope simply to look at things just for the fun of it, because it's a little bit like not only for educational reasons but also for fun. And what I read, and that's also quite nice, I read that even some very unexpected people like composers, for example, all of you know Mozart and his father. He was also a very well-known composer, Leopold Mozart, and in his legacy there were not only music instruments, but there was also a microscope. Yeah, so he was a musician, right? So this is a thistle bug. I cannot read. What is this? Inges? I cannot read this here. Okay, but this is a bug. A bug? Well, okay. Well, let's have a look. Okay, and this also seems to be a very nicely prepared specimen. And this thistle bug. Where is this thistle bug and larvae? There is no information on who made this. Okay, so I do not know. Okay, so let's have a look here. So, but from the writing and so on, I also can imagine that yeah, it is quite an old slide. So here we go again. Okay, that is our thistle bug. Again, a little bit of a look how nicely placed it is. Yeah, yeah, all of the legs nicely oriented, the wings nicely stretched, and quite nice. Yeah, the exoskeleton, which is the outside of the insect, is has been bleached in most places, so it's mostly transparent. You can actually bleach them by submersing the insect in a strong base. Potassium hydroxide is sometimes used, and this causes the insect's exoskeleton to become soft and also brighter. But let's zoom in a little bit here at the head. Okay, again a little bit of a focus. Look at the compound eyes. Interesting. Normally the compound eyes actually look dark. Here they look bright. Okay. Quite a nice specimen as well. The abdomen. But it could be, I don't know, it could be, just by looking at this, look how flat it is. It could be that whoever prepared the specimen has removed all of the internal organs, or most of the internal organs. Yeah. Simply to make it better visible. Here are the wings. Okay, let's go in here a little bit higher with the magnification. Okay. Yeah, and of course the feet. Again a little bit higher. Let's have a look here. Yep, with the two, yeah, with some claws here. Yeah, so how can people find those antique slides? Well, yeah, with a lot of patience. Yeah, and yeah, I know there are some. I've seen some web pages of some people who were selling those. Yeah, so yeah, that's basically, and some of those slides can be quite expensive. So this is a little bit a thing as well, that some slides are collector's items. I'm not so much into, I don't have a lot of knowledge, so I cannot decide whether some slides are valuable or not. Okay, so but that is essentially something that yeah, one simply has to go a little bit like collecting postage stamps maybe. Yeah, but also quite nice here. I'm quite happy with this specimen as well. Almost looks like another being growing on the foot. Yeah. Yeah. So let's go down out a little bit again with the magnification. And maybe it could be that those structures here, also the trachea. I don't know. Yep. Okay. Let's go on again. I want to show you. Oh, look, it's also pretty cool. Here, yeah, here are the eyes. Again, as I mentioned before, a little bit unusual that the two eyes look that bright. Normally the eyes look dark, but this must have been brightened up by the chemical treatment of the insect before it was mounted. So let's move on. Let's move on again. I have two more slides, and then we can have a quick look at the end, maybe at some of the two specimens that I made last week. So let's move this away again. So what is this here? This is larva of a tortoise beetle. The larva of a beetle. Okay, larva of a tortoise beetle. So let's have a look here. So that's basically the larval stage. Insects, of course, have a larval stage as well, and then they undergo metamorphosis to form then the adult. Okay. Let's remove the polarization here. Oh, wow. Okay. Wow. Okay, that's also a very thin specimen. Look at this here. Let's go down here as well. Lots of spikes. This is definitely not a, it's also a compressed one. So this also seems to be mostly the skin. Okay, so, but the mouth parts also seem to be quite interesting here. There's a little bit of dirt here. So let's open up this here. Yeah. Yeah, these are scary looking mouth parts. So, the mandibles, as we call them. Yeah. That's the larva. This larva seems to be quite aggressive as well, huh? Look at all those. Yeah. And just looking at it is very clear that this is this specimen. Yeah, the organs were also removed, so that there's only the outside left over, so that it's actually possible to mount it properly on a slide. Yeah, a little bit of yeah, some dust fibers, and very spiky. Okay, here the feet, maybe these look like a little bit like feet. Actually, larvae don't have to have feet necessarily all the time, but maybe just some possibilities to anchor them into the ground a little bit to aid in forward movement. Yeah, it's a bit. Yes, some of you are saying it's a scary looking specimen. That's right. I don't know what that is. The brown part here. Yeah, I don't know if it doesn't look like the contents of. Nah, I don't know. Maybe it's something external on the slide. I don't know so much. Yeah. Yeah, but that is in any case. This specimen in any case has also survived around 100 years. Looks like mold. You know, I don't know. Could be, but I mean, then. Yeah. But then I don't know if mold actually spreads like this. But then of course you never know. Could be. I don't know. Yeah. I would be a little bit surprised if it were moldy, because if they actually used some kind of a mounting medium like Canada balsam, I mean, then mold would not be able to grow. Yeah? No moisture present. Yeah? How would someone remove the organs from something so tiny? There is a YouTube channel by someone who is preparing insects. Unfortunately, I don't know the name of the YouTube channel. And he actually shows you how he's preparing those insects. And what he's doing is the following. In this case of the spider that he mounted, he basically compressed it, and the spider, you cut it open on one place and you just squeeze out all of the organs. Sounds kind of extreme, but essentially it's not necessary that you actually dissect it like this, but you just make a small hole and you carefully squeeze out the organs that have become soft during the preparation, and what's left over is just the outside part. Yeah? So this is, yeah, I don't know, okay. So what I'm going to do now is, I'm going to move on to the next one, because I'm already talking over an hour. And let's have a look at another one. The last slide is a cat. And here it is. Okay. This one here, let me check. The Mesentery of a Cat is also by W. Watson. Okay, it's also one of those. Oh, look. Here it says W. Watson and Sons, London. Here's the sticker. Okay. The mesentery is basically the tissue that holds the intestines of a mammal in place. So this is basically from the intestine, right? There's a thin tissue there that kind of holds it in place. And yeah, there's a little piece here, and it's from a cat. Okay. So we're going to move on here. And let's put it on here a little bit. And here we go. Let's try to find the. Let's try to find it again. Maybe it's too bright. I don't. Here we go. It's not very darkly stained. Okay. Okay. Could this also? I don't know what these things are. So let's go up a little bit with the magnification again. Yeah. So the cells, individual cells again, are stained. Okay. So here we go. Okay. Yeah, again, the individual cells are visible. Yeah, it's not such an not quite as impressive maybe as the insects maybe, but I think what we're able to see here as well. I don't know if you're able to see this. Again, this fibrous structure. So these again, every time when you see those lines, then very often that's collagen again. That's a very strong protein which makes the tissue very strong. Okay, so I suppose that those tiny lines here, that's again collagen fibers. So the question again, technical question: your DIC detailing is very similar to what I see with M objectives and desk lamp shining from below, but more like crystal dots inside specimen. Is it the same? So when you're saying that you're shining from below and from the side, if light is coming from the side, then this is called oblique illumination. And indeed, oblique illumination looks similar to DIC. In the sense that DIC and oblique illumination, it causes the specimen to appear a little bit three-dimensional because the light seems to come from one side. So as a matter of fact, this is something that where people have also spent a little bit of time making filters and playing around with that, because you can actually sometimes with oblique illumination also see details that you otherwise not be able to see. So it is not the same, but the effect seems to be similar. Okay. You mean the exoskeleton? Yes. Yeah. So I think I'm a little bit, I don't know exactly now to which question this refers to because I already moved on. But this is basically from the tissue or the organ that holds the intestine of a cat. Yeah. And I think that is what we're able to see here are those fibers, which again is the collagen. Again, a slide which is, I don't know, maybe 100 years old. Oh, look at this. Interesting. Oh, Could this be, I don't know, could this be a blood vessel? Could this, I don't know. Maybe this is a blood vessel with the red blood cells. I don't know. Yes, most likely. Yeah. Okay. Actually quite nice. These things in here, they look a little bit like packed red blood cells. Some also from the side. Okay. So maybe these are, yeah. So let's now move on. Well, actually, it was already the last slide I wanted to show you. What time is it? So what I'm going to do now maybe is I'm going to quickly show you the slides because somebody also wanted to know it from last week. Okay. So I'm going to show you again here. Let's put it back in here. So these were basically the 8 slides that I showed you. And what I'm going to do is I'm going to make a separate short video dedicated to each one of those slides, which you can also see, of course, in the in this channel here. Okay. So what I'm going to do now is I'm going to show you this here, which I've showed you. You see this? These are slide labels. Okay, and they got inspired. I got the inspiration from here, and I used a laser cutter. To cut them out, so that's also something that I'm going to experiment around with. But what I would like to do today now is show this is a slide box that I also made with a laser cutter, And in here, I included the slides from last week. Okay, if you remember last week, I made some onion samples. And I mounted one of them in U-Pearl mounting medium, the other one in PVA glue. It's a water-based glue. And let's have a look at those, right? Because somebody was already asking for those. And I'm going to put both of them now under the microscope and we can compare them. So that's onion skin, dried onion skin. And there is a bit of a difference here. Okay. So, I have to, of course, move over here. So that is now the now the onion skin in U-Pearl mounting medium. Okay, and what we're able to see quite nicely. When I change the polarization a little bit now, it doesn't work so much. We are able to see that in here, of course, are those little oxalate crystals. Okay, so that is the one in U-Pearl, and there is the other one, the red one is over here. Okay, this is how it looks like as well. We're able to see quite nicely, yeah, the oxalate crystals which are found in the onion cells. These are the little squares that you see here. Okay, so and now let's check how it looks like with the PVA glue. Okay, this is how it looks with the PVA glue. This was also quite similar in that sense. But notice this here, this is an air bubble. And the thing is that because the onion skin does seem to be a little bit hydrophobic, the hydrophilic, the water-soluble PVA glue does not like to reach all of the parts equally well. So that could be also a consideration here. And we see some air bubbles. Yeah? And let's go down a little bit with the intensity, and I think that the specimen also does not look quite as transparent. So for example, here you are not able to see the individual crystals quite well. Yeah? And compare this now to the one with the U-Pearl mounting medium. Where is this? Yeah. Well, actually, if you compare, there is not so much of a difference, I think. Yeah? But with the red one over here, okay, that's this one, and over here that's the other one. So actually, if you see again here more air bubbles, again I would say that, yeah, I would say that the PVA glue is also fine. Yeah. It depends really very much on the nature of the specimen and how compatible the specimen is with a mounting medium. But if you want to make slides yourself and you want to experiment, then I highly recommend that you just not spend too much money on some fancy mounting media where you need to dehydrate it first, and then there's lots of preparation necessary. But essentially, you just try to use some PVA glue. So I now moved away the filter again. This is one, so maybe we see a little difference here. And this here is the other one. Okay, that's the U-Pearl one. Yeah, it does look a little bit more transparent. Okay, so don't look at your watch. Huh? Yeah, it's 1 hour 17. And what I'm going to slowly do is I'm going to quickly run through the comments again if I basically overlooked something or not. I don't think that I did. But I think what I would like to do now slowly is I would like to simply say slowly again goodbye, and hope that you liked the video. I'm going to, of course, keep on looking around for some interesting specimen slides, and I can also maybe make a little announcement already that there's some people are also sending me slides, so that I'm going to also present them to you, of course. And what I would like to also do is I would like to invite you to please also check the other videos on this very channel. Okay, so maybe there's something interesting there for you as well. And there is a comment here: a cheap solution for pulling a vacuum is using a syringe with a closed nozzle. And as a matter of fact, I bought something today. I bought a huge, huge one, about this size of a pump, which is used for actually declogging the drains of water drains, and this creates quite a bit of a vacuum. And what I would like to do is I would like to also use this for creating a vacuum, so that it's possible to more easily remove those air bubbles. I have not tried it out yet, but I also will report back here. Okay, so what I'm going to do now is I'm going to say bye-bye. Yeah, please do visit again next week. If for whatever reason I'm not able to do it on a Saturday evening, that's my time, then I will make a notification in the community posts. Otherwise, at the same time, I'd like to be here again. I wish you all the best. Yeah. Enjoy microscopy, happy microbe hunting as always, and see you around next time. Bye bye.